I'm going to talk about cancer, and I'm going to talk about how we can try and get our immune systems to recognize and fight cancer in a way that they've been trained to recognize and fight infection. One of the first things to understand is, what is it about cancer that is different to normal cells? So cancer is one word which is used to describe many, many different diseases. And cancer in one part of the body or in one organ of the body can be very different to another part of the body. The treatments that we can use are very different, and the prognosis for the patient, so what actually happens to you, how serious it is, how it affects your lifespan, can be very different for different types of cancers. But there's one thing that's really important and sums up the basic problem with cancer, is that the way our normal cells divide, replicate, and continue to grow through all of our lives, they do this each time a cell divides. It has to copy its genes, and it copies its genes exactly, and so it makes two daughter cells which are exactly the same as the parent cell. And this happens throughout our life, every day. Thousands and thousands and thousands of cells, new cells are being made, and other cells that are getting to the end of their life are dying. So very quietly this happens all the time. And if we make a mistake in copying the gene sequence, so the list of letters that has been shown to you by Siobhan, then we can make a cell that doesn't work properly. And Siobhan's already introduced that fact, that you can make a cell that can't talk to another cell, or you can make a cell that doesn't do the job it's designed to do. In many cancers, what actually happens is, if you acquire a number of different mistakes, that the cell then loses its ability to divide and behave and play nicely. So it becomes like an out-of-control child in a school playground, running around like crazy. It starts dividing very quickly. It's not regulated in the normal fashion, and you end up with a group of cells that behave very differently to the cell that they originally came from. And this is an accumulation of mistakes within the cell. And if the body isn't able to deal with them, this becomes a problem. An example of this, one of the diseases that I'm a doctor looking after, are some of the blood cancers. I look after patients with leukemias and lymphomas. And what you can see here is a picture of, I don't think that, that isn't a pointer. What you can see here is a picture of a normal bone marrow, and Siobhan is, again, you're like a perfect warm-up act, has already shown you a picture of a bone marrow where all the immune cells are made. And you can see here there's lots of different types of cells. This is a normal, healthy bone marrow. And if a mistake is made in a cell so that it suddenly starts dividing, and it divides much, much more quickly, and you suddenly get an accumulation of a whole load of cells with the same mistake in them, you can develop a form of cancer. And this particular example is a cancer of a blood cell, so it's an acute leukemia. And these are two identical biopsies from bone marrow of a normal person and a person with acute leukemia. And it's very obvious to everyone that there's a difference there, that you've got a whole load of one type of cell instead of a mixture of lots of different types of cells. Now, you've learnt a little bit today about the cells of our immune system, and you've learnt about T cells, which are one of the types of white blood cells. And these are the cells that have sensors on their surface, and they're designed to recognize and attack infected cells. And they do this by this particular receptor or sensor that we call a TCR, or a T cell receptor. And they recognize bits of protein on the surface of cells in our body. Now, if you were here at the beginning of the afternoon, you will have heard Lucy talking about diabetes, and about how normal T cells in our body can attack healthy cells in the pancreas by making a mistake and thinking that normal proteins on the surface of the pancreas should be attacked. That's when this system has gone wrong. So in a healthy person, our immune cells will ignore our normal tissues because we don't want our own immune system to start attacking our healthy cells. We do want our immune cells to be able to attack and kill infected cells. If we pick up a virus, we want any cell in our body that's harboring the virus to be killed because that's how we clear the viral infection. Okay? Now, our immune cells are, except for when you've got an autoimmune disease, which you've heard about today, are quite good at ignoring our normal cells. They're very good at killing virally infected cells, unless you're missing a sensor, as described by Siobhan. So, are they any good at killing cancer cells? Well, the problem with cancer cells is actually they might be dividing like crazy, and they might be behaving badly, and they might be in the wrong part of the body, but they look often quite a lot like normal cells in our body. And a lot of the proteins on the surface are exactly the same as the proteins on the surface of a normal cell. So our immune cells that are trained to ignore our normal healthy cells actually will often ignore cancer cells. So instead of seeing them as something dangerous or something different, they see them as something that's pretty similar to a normal cell, and they've grown up knowing that they mustn't attack the normal cells in our body. So how can we do something about this? Is there any way that we can make these immune cells more efficient and more able to recognize cancer cells? So we can try, and Marla introduced this earlier when she was talking about the gene therapy treatment for the gentleman with the hepatitis B and hepatocellular cancer. We can equip our immune cells with a new weapon, with a new T cell receptor. And we can do, if we do this and we're successful, we can actually change what the immune cell recognizes. And sometimes, and some of the work that we're doing in the laboratory is we're trying to see not only can we change what the immune cell recognizes, so if we've got an immune cell that maybe recognizes a virus, can we change it so it can recognize a cancer protein? But can we also change the immune cell to make it a better killer cell, to make it more efficient at killing the cell that it recognizes? And can we even tell the immune cell which part of the body it needs to go to? Can we actually redirect it? Which is important because some cancers are in parts of the body that it's quite hard for the immune cells to get to. So how do we do this? How do we sort of give new receptors or new proteins or new functions to cells? And one of the things we have to do is deliver the genes that provide the instructions for making the receptors. And the genes are delivered to a cell using, surprisingly or not surprisingly, one of the things of the world that is very good at getting inside cells, and that's a virus. So actually we're sort of being a bit, this is a slightly out of focus picture, and these blobs here are viruses, and they're getting into a cell and infecting the cell. So although we usually think of viruses as being our enemies, actually viruses are brilliantly efficient at getting inside cells because that's where they live and that's where they divide. So if we can take a virus and use it actually as a method of delivering new genes to a cell, we can change the genes inside the cell. So how do we do it? We take the genes that give the instructions for making the new receptor, and we put these genes inside a bunch of cells that we have growing in our lab. They're sitting in a little plastic dish in the lab, and we add these genes, which are in a sort of well, it just looks like a tiny bit of fluid, and we add it to a dish of cells. And we grow these in the lab for a couple of days, and these cells are able to make a virus that's not a real virus. It can't cause an infection, but it contains with it, inside it, the genes that we've added at the beginning. So these packaging cells leak out this virus into the liquid that surrounds them in a dish. Okay? So we take this liquid, or supernatant, if you want to be fancy, which contains these virus particles. These are non-dividing viruses, but they are able to deliver the genes that we put in at the start of the experiment. So we then take this liquid, or this soup with the virus in, and we actually then mix it together with a whole load of immune cells, or T cells, in another dish. And if this virus does its job properly, it gets inside the immune cells and delivers. It's a bit like a fantastic Amazon courier. It delivers in 24 hours your gene that you've asked it to deliver to the inside of the T cell. And you can just do that in a dish. And you're not doing anything fancy. You're just asking a virus to do what a virus does. And a virus gets inside cells. But it's a great way of delivering something into a cell in a natural way, rather than trying to poke a hole in a cell without killing it and squirt something in, which of course is impossible to do because they're so tiny. Although if anyone was here to listen to Alex's talk, you wouldn't believe anything was impossible anymore. So these immune cells have now got inside them the genes which allow them to have the instructions to make the new receptor. And they make the new receptor, and that gets put on the surface of these immune cells. So you've basically, in the course of a week in a lab, you've created a bunch of immune cells that all have the same receptor, that can all recognize the same thing. And for our particular work, what we're trying to do is make groups of immune cells that can recognize cancer proteins on the surface of cancer cells in the body. So then we can take these immune cells and infuse them back into patients and ask them if they can recognize and kill a cancer cell in the patient in the same way that we expect our immune cells to recognize and kill virally infected cells in their normal course of events. Okay, so this is another picture. This is what I talked to you about before. If you have a normal, unmodified immune cell, it looks at the cancer cell. There's a lot of proteins on the surface that look quite similar to a normal cell, and the immune cell goes, no, I don't think I'll bother attacking you. But we use one of our gene-modified T cells, which has got a new receptor, which is very powerful at recognizing all these proteins on the surface of the cell. This new gene-modified immune cell is able to kill that cancer cell. Okay? So we've done a number of experiments, and if you do that, that would be perfect. And this experiment here looks like a bunch of bars to you, which is exactly what it is. This is the name of a chemical that is released by an immune cell when it's activated. So when an immune cell sees something that it recognizes, it releases a chemical called interferon gamma, and that helps kill the target cell. And what you can see here is if you take some immune cells from some patients or some healthy volunteers, and you put into them the new T cell receptor using our gene therapy approach that I've described, and then you mix them up either with a cancer cell or with what we call a positive control cell, which is like a designer cancer cell, or a cell that we would expect our modified immune cells to ignore. You can see that our gene-modified immune cells can recognize the cancer cell, so they squirt out this interferon gamma. You can see a toolbar. They can recognize the positive control cells, which are the dummy cancer cells, but they ignore the negative control cells. So we can tell that putting our new receptor on the surface of the immune cell actually makes them quite good at recognizing cancer cells in a dish. And the immune cells are able to recognize them and secrete this chemical, which is their normal response to a target cell. What this plot here shows, that's okay, it's all very well doing an experiment in a dish because you've actually put your immune cells sitting right next to the cancer cells in a dish. If your immune cells can't do that, they're not going to be any good to anybody. Okay, you're making it pretty easy for the immune cells, because they're just stuffed in a dish with a whole load of cancer cells. This experiment here is an experiment that we performed in the lab where we took some mice that had been injected with cancer cells, and we then infused into the mice the gene-modified immune cells. And the reason that we do these experiments is that we have to see whether if you inject the immune cells into the bloodstream, can they get to where the cancer is? And when they get there, are they able to work? And are they able to recognize the cancer cells? And are they able to kill the cancer cells? We cannot get any of that information from a dish. And we also need to do these experiments so we can find out whether the immune cells cause any damage in the body when they're injected. So the next slide. These are some mice in our laboratory. They're anesthetized, they're sleeping, and these fluorescent blobs on their backs are tumor cells growing under their skin. And these mice have been treated with normal immune cells. These are immune cells that we haven't done anything to in the lab. And what you can see over the course of the time that the tumor cells are getting bigger. The immune cells are not able to recognize, kill those tumor cells, or make the cancers go away in these mice. But if we do the same experiment in the same mice with our gene-modified immune cells, you can see here beautifully, and this is much more, it's a much convincing way to look at the effect of our immune cells than looking at a dot on a graph. You can see here that the cancers that were growing at the same rate at the beginning are being recognized and killed, and the cancer cells are completely disappearing in these mice. And these immune cells, we haven't injected them into the tumor, we've injected them into a tiny little vein at the end of the tail of these mice. In the same way that if you're a patient and you go to a hospital and you get a drug, it's injected into a cannula in a vein in your arm, and it's got to get to the right part of your body. So this is a very good mimic of the sort of treatment that we're planning to use in patients. So we have some clinical trials that we are beginning to recruit to using gene-modified immune cells. We have a trial that's open where we're treating patients with leukemia. We have another trial that's open where we're treating patients who've undergone bone marrow transplantation, where we're hoping that our gene-modified immune cells will help prevent people getting serious viral infections after their transplant. We are planning another study which will target two types of cancer at the same time, nasopharyngeal or throat and nose cancer, and a particular type of lymphoma. And these studies are open that's UCLH down the road by Euston, and this of course is here at the Royal Free. And our colleagues and collaborators, also part of UCL, but at Great Ormond Street for Hospital, have a lab where they do the gene engineering of immune cells, and we go down and do that with them. We work together. And this is the lab there that did some of the original gene therapy studies for children with immune deficiencies. So we've built up a strong partnership with them, and that's where we make our gene-modified immune cells. And this is what actually happens. If you're a patient and you're going to have this sort of treatment, you go along to clinic, you get attached to a machine called an apheresis machine, and some of your blood cells are collected from your circulation while you sit reading a magazine or watching the telly for about 4 hours. And these immune cells get couriered to Great Ormond Street, and then they're processed in a very clean laboratory called a GMP lab for about 7 days, and they get modified in the way that I described on that diagram. And then you end up, these are your starting immune cells, and at the end of the week in the lab, you've got, these are your immune cells. And what you can see here is we've been able to make a group of immune cells that all have the same receptor on their surface. And these immune cells can all recognize a cancer protein. Okay, so we've started treating patients. This is the bag of immune cells that you start off with. This is what you end up with at the end of the week. This is the first dose of gene-modified T cells. That, for your own records, is a vial that's about this big. So it's probably, in old money, it's an inch high. In new money, it's about 2 centimeters. And the volume of cells infused is about 1.5 mils. So for the first patients treated, we're giving a number of immune cells, we're giving between 1,000 and 10,000 immune cells. That's it. When you think how many billions of cells there are in our body, it's a tiny dose that we start off with. And that's how you infuse them into a patient. And that's our funding bodies who've made this all possible. And so by way of summary, it's a very exciting time for all of us because working together, we can understand so many different aspects of how our immune system works. And we can share information about the basic science of how immune cells recognize target cells and do it efficiently. We can also understand why the ways in which they recognize cells we don't want them to and how we can stop that happening. And we're also, by working closely with people who are interested in studying viruses, we're able to use their science to help us change immune cells in the way we want to help them treat things like arthritis, hepatitis, associated cancers, and other cancers. The Royal Free and UCL and UCL's associated hospitals, we're one of the first centres in Europe doing these kind of gene and cell therapies. It's a really exciting time, and Hans has been at the forefront of helping build this new institute, and it's very exciting for all of us who work here. And we really hope it will be very exciting for all our patients in our clinics, because we'll be able to offer you these new therapies to you. And I think the next 10, 20 years will be a great time to be kicking around the Royal Free. Thank you.